Welcome to Life, the Universe, and Hot Dogs. I'm Patricia Princehouse, Associate Director of the Institute for the Science of Origins, and I have a nice red wine here. And I think Jurgen, our speaker, what did you have? Hold up your, he has a local resurrection beer. Let me just say a couple things first here. So this is part of our new Origins Online, which is for the foreseeable future going to be our main mode of communication. We have Life, the Universe, and Hot Dogs Tuesdays until the 14th, Thursdays and Saturdays indefinitely. And then starting on the 14th, we have the Origins Science Scholars every Tuesday for 2 months. You can, of course, find our videos anytime on Origins.case.edu from past Origins Science Scholars program. And a reminder that this coming Thursday, we'll have Life, the Universe, and Hot Dogs with Glenn Starkman, Director of the Institute, on what's hot in cosmology right now. So that's at 7:30 this coming Thursday. Oh yes, this Origins Science Scholars this semester we'll have 2 talks by Dustin Tyler. We'll have Jurgen giving us a longer talk on the corona pandemic. Mother and Child Health with Nikki Burt from the Cleveland Museum of Natural History. Fire, Food, and Our Future Climate with Chris Cullis. Smart and Sensible Renewable Energy with Bob Savinel. And Johannes Haile Selassie from the museum is going to talk to us about the new skull that he found in Ethiopia, so that's exciting. So we're going to go to our speaker here. Just a couple Zoom tips for those who are a little new to it. Keep your mic on mute, and you might want to keep your video on mute except for questions if you're having bandwidth issues. You can hold the questions till the end or until invited. Jurgen may periodically ask if there are questions. Once you want to ask a question, just unmute yourself and ask the question. If there are overlapping ones, we'll call, but generally it works out. If you are having trouble seeing the PowerPoint at the top of your screen, you should be able to adjust the PowerPoint size or the presentation size, which is a keynote in this case. And just put it on 50%, and you'll be able to see both the speaker and the PowerPoint. Okay, so Jurgen, if you want to put your...
Just one thing, Patricia, it really does, I noticed not everyone's shut their videos. It really does help everyone if you shut your video.
It's kind of nice to see people though.
You have to end sharing the screen, I think, before I can share my screen.
Oh, it should tell you, okay. Go ahead.
No, now I can.
Excellent.
Okay, so let's just get rid of that. So I think everybody can see my slides now. Patricia, can you briefly confirm?
I can see them.
Okay, perfect. Okay, so today's talk will be about coronavirus, and this image is fairly complicated, and we'll talk a little bit about the different parts of this image at a later point. But it's a very beautiful image, and at the very last slide shows who the actual artist behind that image is. First of all, I want to clarify that the virus is called SARS-CoV-2 for Severe Acute Respiratory Syndrome Coronavirus 2. And the disease is called Coronavirus Disease 2019 COVID-19. So these are 2 different things. The disease is caused by the virus, just to keep those apart. So people keep mixing up certain things every now and then. And it is indeed very famous for this beer. But the reason why I put this in is because you have this little crown in here. So the name of the virus stems actually because it has a crown-like structure, and that can be seen more in this rendering here. So in electron micrographs, for example, when you stain those virus particles, then they stain in such a way that it looks like a crown, basically. And coronaviruses are single-stranded RNA viruses. They were discovered in the 1960s. And they are usually a cause for many respiratory infections in humans. There are a couple of common cold strains that we have on a regular basis, basically every year, and they're called 229E and OC43. These are very common, but they don't cause a very severe disease, like just a common cold, basically. But then there is this whole bunch of other coronaviruses that are also causing disease in humans, and these are more the serious ones. For example, 2010-11, we had the first SARS virus. It was only called SARS at that point. Now they start naming it SARS-CoV-1 or sometimes just SARS-CoV to distinguish it from the current one. Then we also have one that it's called human coronavirus, and then several strains, NL63, HQ1, and then the MERS strain that came from camels, for example. Now the latest beast on the block was initially named 2019 nCoV for new coronavirus, and it then became later known as SARS-CoV-2. And I'll try to refer mostly either to CoV-2 or SARS-CoV-2, depending how lazy I get. But that's the virus we will be talking about today that's causing the COVID-19 disease. So let's start out with a slide that I showed on the 29th of January in a lecture that I'm giving at CASE. And I thought at that time it was fun to include some very new data to expose the students to something that's going on. At that time, nobody really had an idea that this would turn into a pandemic. But let me just walk you very briefly through this graph. So what you see here on the x-axis are days. And on the y-axis, on a logarithmic scale, you have the number of confirmed cases. And this is in China. So we know that there were earlier Chinese cases in December. But the first time when data became really publicly widely available was the 21st of January. So I started collecting the data since then. And on the 29th of January, I gave a lecture. And basically just doing a regression curve, you see that this is a fairly nice fit. And you could predict how many infected cases there should be after a certain time. Now, you see that these lines that came later, they don't follow that red line. So what China did, and actually it did this already on the 22nd of January, is they shut down Wuhan. And so it took a couple of days before it had an effect, but you see here that the curve flattens. That's what everybody keeps talking about, flatten the curve. And you see that the original projection where it would have been has declined significantly, and it further declined. Just as a comparison, this green and red line show the cases that we had with SARS-1 in 2010. So we massively exceeded the amount of cases with SARS-CoV-2 now. Now, how is the US doing? And this is more recent data from March 8 of this month. And we only have about 10 data points that I plotted here and made a progression basically. And so the prediction would be April 1st, which is tomorrow, we would hit about this level. Now, since we have this very convenient web page that is hosted at Hopkins and that the world basically gets the data from, we can look today what the levels are. And it turns out that in US, we actually have way more cases than what the prediction would have been, which means that we're not slowing the curve at this point, we are increasing the trend. Same with the death rate. Now let's go a little bit back to the history here. So the very first case in US was in January 21st, and that was a 30-year-old traveler from China. He died in the state of Washington. On the 23rd of January, there was this massive travel ban put in place in Wuhan. So basically you're shutting down a city of 11 million inhabitants and nobody is allowed to go anywhere. And shortly thereafter, they started constructing these 1,000-bed hospital that you probably all have seen because they were aware what disaster was heading towards them. So basically in the US and European citizens were evacuated in that week from China, brought back to military bases, for example, to monitor them for 14 days and to ensure that they don't spread the virus eventually. And then the WHO finally declared a global emergency. They probably should have started declaring a pandemic already at this point, but a pandemic per definition is only if a disease occurs on multiple continents at the same time. So it was a little bit bean picking, I would say. Then finally, mid of February, the WHO declares COVID-19 as the official name and not the strain from China, Wuhan, or whatever. It's COVID-19, that's it, and SARS-CoV-2, nothing else. 22nd of February, we had the first death occur in Italy, and the first death a week later in US. And then already beginning of March, basically, first week of March, Wuhan closed their makeshift hospital because they didn't need it anymore. So what this basically means is January 23rd, China had about 500 cases, so that's down here. And they basically locked down the city for 6 to 7 weeks and then went almost back to normal. I was Zooming with a former classmate of mine last weekend who stayed in China during that whole time. And he just said that last Friday, he went the first time into a restaurant. So they are releasing step-by-step ways how people can get together again. But it's that amount of time that it will take if we follow the rules that are in place in some areas. Now, if you think about this virus, you can also think about air travel. And this is a map that's basically showing all the non-stop flights out of Cleveland. You can also think of it the other way around. These are all places from where the virus could come to Cleveland. And that's basically what's shown then on the next map. So this is a map that shows the distribution of the virus to the different areas of the world. And what one can clearly see is the larger, for example, a circle is, the more cases you have. And we'll just start here in Asia, China. So you see that there are these very thick branches and thinner branches. So thicker branches means there was more outflow into other regions, in this case, for example, to Europe, or if we go in this direction, to the United States. And you can see that the different colors mean from which country do the viruses originate from and spread into which countries. So there is a nice back and forth connection between US and Canada, which is expected because we have trade with them and so on. But there's also quite a significant amount of viruses coming back to Europe as well. And so far, the African continent has been, let's say, less frequented, but it's just a time point because there is less travel. Certain areas in Africa, for example, those places that see more tourists have more incidence rates, and so it will spread all over as well. What you see here is basically a family tree, and we don't need to go into much detail other than to say that this red branch is basically US. And what you have here is you can align the genome of the virus and define where it came from, because they're all related to each other, because every virus makes little mistakes when they're copying their RNA. And the small differences in the RNA sequences can be traced back to make such an alignment. Now, what you also see in the next slide is all the red dots are American or viruses that can be traced back to an American origin. But for example, the purple areas are Asian countries, the green areas are European countries. What this means is basically that US has exported some of the coronavirus strains back to other places in the world. And in fact, the very first case here, the Washington State one that died, is this point here. And so all the sequences that are US type go back basically to this one ancestor. And so it's very interesting that one can do this. And that's the power of modern technology. So there are over 2,500 genomes of SARS-CoV-2 currently available. And there are daily more added into those databases. Now, looking at a specific area of the genome, and this is basically the whole genome of the virus, it's about 30 kilobases. There are several proteins that are encoded here in different colors. The one that's of interest to us now is the so-called spike protein. That's this green guy. And you see here these high bars, they indicate differences in the sequences. So not every virus will have the same signature. You can think of it as a fingerprint. So every virus has a distinguishable fingerprint from different families. And so with that, you can basically trace back where that virus came from. Now, looking at this spike protein in the area that I just highlighted here, it's here again. It's the interesting area where the spike protein interacts with the human receptor protein called ACE2. And the receptor binding domain that's indicated with this bar here in teal or whatever that color is, it is fairly well conserved between the different SARS variants. It's also related to bat SARS or pangolins. So the interesting thing about where did this virus came from, it can be traced back through sequences. And there was for a long time on Facebook or Twitter messages that this virus emerged out of a laboratory experiment. But having sequences in nature that occur naturally basically demystified that wrong information. Now, when we are infected with the virus, we generate antibodies. That's a normal procedure. Unfortunately, some people don't generate antibodies fast enough and they then become sick. But neutralizing antibodies that would basically block the virus from progressing into your cells would have to be directed against this receptor binding domain where ACE2 binds to. Now, during my lecture that I gave in January, I was able to access the very first sequence that was available from the coronavirus because Chinese researchers made it freely available on January 8. And what I did there basically is looked at 3 different proteins and they all aligned back to bats basically. And Austin, who is also here in the audience, did this whole genome blast and same results. All things pointed towards that virus originated most likely from bats and then jumped from bats to humans. I don't know if you can read this small, tiny little letter, but these are about 4,000 cases. That was January 29. It seems a long time ago. Now we have 20-fold more cases, 200-fold, 800,000 is what we have now. Now, there are also already several publications out there that basically say the same thing. So SARS-CoV-2 and SARS-CoV-1 are closely related and they both are closely related to bats. So you have all these guys are all bat, similar to bat coronaviruses. And I should say that is not surprising because in bats, you can find a ton of coronaviruses. There are about 300 different strains that are known to be in bats, but they rarely transmit to humans. Now, a big guy who is probably one of the best coronavirus experts, he works at the Charité in Berlin, Christian Drosten. He has a podcast, which is unfortunately in German, but it's extremely informative. And so this is one of his papers actually that he published. Coming back to that sequence that I was saying, one big difference that is not present in any of the known sequences that have been identified so far is this so-called polybasic cleavage site. So one thought is that probably that insert happened while the virus was already in a human. But that's now the sequence that's present in all viruses. So it might have been early on that there was not such a sequence and maybe not such severe cases, but over time it mutated into having this insertion in that region that may have made it behaving differently. We don't know for sure at this point. Now let's have a brief look at how the disease progression happens. And I've made here a very, very simplified schematic, but I think it helps to drive the points home and to introduce several terminology that is important for you. So first of all, here on the x-axis, we have the number of days arbitrarily. I set a sore throat as one of the, let's say, known recognizable symptoms of COVID-19 as day 0. And then you have on the y-axis how much viruses you have in your throat or lungs or whatever. Now what happens at the beginning is that you get infected at some point. The virus replicates in your throat, and at some point you feel, okay, there is something wrong with my throat, I'm getting sick here. And then you peak at some point. And then the throat virus decreases again. Now, if you make a COVID-19 test and you test either early, because you just want to know if you're sick or not, or very late, you get a negative result. And the reason is simply because there are not enough viruses that can be sampled at that point in time. Now, what really happens is at the same time, while you have the virus developing in the throat, you always swallow and you breathe in, and some of the virus gets into your lungs. So it's also developing in the lung at the same time. And it peaks at some point. So the viral load in the lung usually gets much higher than in the throat. Now, if you have someone who is terminally ill of this disease, the virus just continues growing exponentially and you eventually die because you cannot be saved. But we also have methods of prevention, for example, putting someone on a ventilator or potentially with drug treatment. There are other methods around that are being tested right now, like taking serum from convalescent patients. And I can talk about that later as well. But basically, you rescue somebody who is on the track of getting really sick to combat the disease, basically. Now, the interesting and tricky part about this virus is when are you infectious? And that is really nasty and not nice, because there are not many viruses where you are already contagious to others before you have any symptoms. So if we compare this to regular flu, typically a day earlier before you really feel ill, you know, yeah, I caught something, right? So you stay at home. And that's the tricky part with this virus. You don't know that you're sick because the first signs of sickness appear about 2 to 3 days after you've been already able to transmit the virus. And that's, so basically this area here before left of the 0 is the critical part. And that's what people don't get. Like if you get together with people who are asymptomatic, they may have the virus and transmit it to you. And that unfortunately happened recently in a choir in Washington State. 60 people were performing, I don't know what they were performing, but they decided they would continue performing their things. And they all tested, or all were asymptomatic, but it turns out that some of them were actually sick, and now 10 of the 60 died already. So that was pretty bad. Now, in terms of tests, so I already mentioned that if you test at the wrong time for the throat or nose swab, you will get a negative result even though you have the virus. And that's indicated with this color intensity, basically. So at the peak, you have the highest viral load, and then it's the easiest to detect. If you were to do a lung bronchoscopy, you could detect the virus early on until late, but that's not a normal procedure that people voluntarily undergo, but you could do that and detect the virus. The other option is to take stool samples. Now, it has been reported that the virus has been identified in stool, and that is correct. And so basically, as soon as you are infected, you can shed basically the virus. And all of these 3 types of samples can be tested by so-called polymerase chain reaction or real-time PCR. I'm not going to go into the details what they are exactly, but it's a method that allows you to amplify the viral genome in a short period of time, let's say about 4 hours, and you get a result. Now, the reason why you then don't get the result back within 4 hours, if you run or go through one of these COVID drive-thrus, is because you typically run this in a 96 well plate and you wait until all samples are there and then you run the sample. So it's typically once a day you run several plates or something like that. So it usually takes a day to get the result. In US, we've had the problem that many of those tests did not come back within a day. Several people got results back 8 days later. So it's a problem which I think is being addressed now in a better way. There's another way how you can test infectivity, and that's called a plaque test. What you basically do there is you have the virus particle and you dilute it in a serial dilution, and then you plate it on a human cell tissue culture, basically. And you look how many cells lyse with whatever concentration of the virus you have until you don't see any lysis anymore. So that's determining basically how many virus you have in your plate. And you can do that again with all the different samples. And basically only for the throat and nose swab and the lung bronchoscopy, you can get viable parasites. The stool sample that is positive with the PCR test, indicating that there are virus particles in there, cannot be grown. So that means that the stool sample viruses are not infectious. You can use the same assay basically to do drug screening. And in that case, what you do is you change not the virus amount, but you change the concentration of the drug. And this is then indicated here as an example where you have 3 drugs. The black drug is not very effective compared to the red drug. And you can determine certain values that are important and an indication for how good those drugs are for a particular assay that you're using. 2 values are important, EC50 and EC90. And the EC90 basically describes reduction of 90% of the infectious particles. So typically you would like to look for these values to be more effective and more sure that you kill most of it. Now coming back to our simple schematic here, there's another thing that you can do, but you can only do this after the infection. And this is a so-called antibody test. So, and this is very important for those people who, let's say, were asymptomatic. Like if you had the virus infection, but for whatever reason, you only had a very, very mild symptom and you didn't even care to go for testing. So you don't know that you were sick. You still generated antibodies. About 2 to 3 weeks after an infection, your immune system starts generating antibodies, and these can be tested. And there are in principle 2 tests that you can do. So the only thing that you need is a blood sample from someone who suspects that they were infected. And you can either run a rapid diagnosis test that is very similar to what is in place for malaria diagnostic and other things. And the principle is very simple. So the blood that you pick up here is put into this rectangular box, and it's sucked up into a paper that's here. And you put a running buffer in here that lyses the red cells and moves everything over here and frees basically, in the malaria case, the parasites. For the virus, it would free up the virus particles. And then you would get antibodies that are already in this, on this paper-based assay, would capture the virus proteins and would indicate if there is virus present or not. And you always have a control that you know that the assay actually worked. The other type of assay that people can do is a so-called ELISA titer determination. And what you do there is you coat the plate with virus proteins. And this highlighted antibody is basically your serum from the patient. Now, if you had the disease, you will recognize the virus particle or the virus proteins that are there, and your antibody will bind to that protein. Now you need a secondary antibody that can recognize the human antibody because we need to somehow detect it with a signal. And the best way of doing this is to detect the fluorescent signal that is indicated here with the yellow. And so you can basically read out, yes, that patient has antibodies that react against virus proteins, and you can even determine to what degree it reacts against virus proteins. Now, you probably all have been following the Ohio state official dashboard on COVID-19, and this is as of yesterday. I forgot to update that slide, I'm sorry, but I should have, I don't know where we are now with today's numbers, but it is very informative because it gives you where in Ohio and to what degree do you have cases, and obviously Cleveland and any area that is highly populated will have the highest amount of cases. That's just normal. And you have basically the total cases, how many are hospitalized, and unfortunately, or how many are dead. Now, one thing that Ohio does differently than others is, and it's not on their dashboard directly, but you can download the data and you can see how long, when was somebody submitted for the hospital and when did they either leave or die. So you can look at very, very detailed information if you wish to do so. I've been going to this webpage way too often, but basically to look at the new data, download new data, and do some analysis. And you can see here, US is leading by far. Probably in 2 or 3 days will exceed 1 million cases worldwide. And what is interesting, if you look at the distribution of the red dots, you see that there are some creeping up in Africa. If we would zoom into, you would see every single African country has a few cases. They are about 3 weeks behind the US. So there will be devastating scenarios in Africa as well. So where do we have to be is the question. Now, here on the left graph, what I plotted here is the data of the last couple of weeks. And this is a different type of plot compared to what other people show. And what it's shown here on the y-axis is the doubling time. So basically the doubling time tells you from, let's say, today, how many days does it take to double the number of confirmed cases? And the larger the number, the better it is because it's slower. So that's again flattening the curve basically. Now, if we look at China here in blue, you see that, and this is shifted so that we can put it on the same graph, but this was technically earlier on. But if we compare their data, so basically here on between weeks 6 and 7, they instituted the travel ban in Wuhan. And there was not much seen for a couple of weeks, but then all of a sudden, everything stopped basically. There was no progression of the virus transmission. Now, Italy, the green line, oh, sorry, this is South Korea. South Korea, the green line, they have a slightly different approach, but they are also on an extremely well maintained way of controlling the disease. Italy here in red is slowly taking the curve up. So hopefully they will continue. I mean, it is very likely that they will continue to improve. But if you look at all these US states are plotted here, they are all down in the weeds. So we are not doing very well. The only ones that are comparable to Italy now are the Virgin Islands and the District of Columbia. So this is not too good news. And we've instituted in some states, I think around week 7, between week 7 and 8 is, no, actually 8 is where we instituted certain stay-at-home orders. As of yesterday, Maryland has actually a fine of $5,000 and 1 year prison if you are caught outside with no good reason. And I think that has to be done basically for all the states if we want to be successful in preventing this disease from spreading. If we compare how the whole the whole range of data that we have for Italy and just New York City in this case. So Italy is this first curve more to the left, and the doubling rates are here in this box for this. So at the beginning, basically in less than a day, the cases were doubling in Italy, and then it started slowing down slightly. Right now we are at about 10 days doubling time, so that's not good yet, but it's better. In New York, however, we are not doing very well, and the New Yorkers know that. I mean, that's obvious. But we also see a slight trend that it is already bending the curve here. So hopefully this will, they will be able to maintain that and be better at it. New York alone accounts basically for 43% of all cases in the US. That's something to keep in mind. So basically, you can directly compare New York to Italy, the whole country at this point. Now, what do you do when you have symptoms? And that's, I think, one of the important parts, because it's a little bit unclear what symptoms are influenza, what is a normal flu, what is allergy, and so on. But I think what is very clear is if you have these three symptoms at the top, you cough, you have fever, and you have shortness of breath, that should be a slight alarm signal for you to at least investigate. I should seek out potentially some medical guidance and don't wait with it, because in particular in Ohio, I've seen, as I mentioned before, that you can go to that website and get every data that you want. I've seen 3 or 4 cases where the people were brought into the hospital and then died the next day. And the reason for that is basically they waited too long. So now the best ways of protecting yourself is wash your hand. We know that in the meantime. Sneeze into a tissue, hold try to avoid aerosolizing stuff, and don't touch your face with your fingers like I'm doing all the time. I washed my hands before. If you have somebody in the household who you suspect is maybe exposed, isolate. And I mean, that's in particular for our healthcare workers. I mean, many of the people who are in the healthcare industry, if they can, what they've done, let's say if the partner doesn't work in the health industry, they would just divide up the house. One sleeps in the basement or in the attic, just to avoid contact and not to endanger the families, basically. That's one important way of separating yourself. And then, of course, using sterilizing method to wipe down surfaces. And you should only wear masks when you're actually sick, to protect other people from getting like droplet infection. It's not so much protecting one from others. It's really more you protecting others from your sickness. So health professionals need to wear these masks. So don't buy them and take them away from them, because they are in the front line and really are exposed to way higher viral concentrations than any one of us at any normal day. Even if you go shopping, it's much less exposure than those guys in the hospitals. They really need them. There are several pieces of information where you can turn yourself to, like the support line 4459 COVID, or also the Department of Health from Ohio. It's a hotline. If you feel you need, or if you feel sick, reach out to those hotlines. Some of them are a little bit overloaded, but it's worthwhile doing this before you are really sick. Now, the last slide that I wanted to share with you again is the first slide that I had. So this is Veronica Hayes. I think she has a middle name in the meantime. She's a medical illustrator student at Johns Hopkins University. And I have not personally worked with her, but I've worked with many people out of this program for several of my illustrations that I have on some of my publications. And this is a really, really very nice summary of what is currently known about SARS-CoV-2 spike protein and the interaction with the human system. So I'm happy to take any questions, and I hope this was informative. and I'm willing to talk about anything else what you want to talk about.
I guess as a question from the audience, if you in your line of expertise were tasked with finding a way of combating this organism, what would be your method of approach?
Oh, there are many methods. First of all, we don't have anything that we can use instantly. Like the only instant, like, how should I say it as? The safest is to stay in house and not expose yourself. Just minimize the risk, wash your hands. And the reason why washing the hands helps is because that virus has a lipid envelope, and basically the soap is solubilizing the lipid. The other thing is, if you don't have the option of staying home and you need to basically go to work and all these things, in that case, just try staying away as far as possible from people. Minimize exposure to gatherings. That's the safest you can do. Now, in terms of treatment options, I think there are different ways how you can define this. I mean, we do have certain ways just with ventilators, no drug, like helping people breathe, it's one option, and decreasing your own immune response to the virus at that point. So one of the problems that happens for these people who are, let's say, really ill, they have a much stronger cytokine reaction that destroys the lung. And basically, if you reduce that cytokine reaction, you reduce the damage of the lung, but at the same time, you have to ventilate them and keep them basically alive and hope that their immune system kicks in to generate the right antibodies to prevent the virus. The thing that I think is on the short term achievable is the covalescent serum approach that got fast-tracked by the FDA to use as a compassionate use approach. What this means is that it's not an FDA-approved drug or treatment, but people who are sick, they have the choice of either dying or agreeing to, yes, maybe this can help. It's basically the same method that was used for the Ebola crisis, where American company produced humanized monoclonal antibodies against Ebola, and it was not tested in animals or anything, so there was no FDA approval. But because people were dying so rapidly in Africa at the rate of 70%, like if you're infected, there was a 70% chance that you will die. So basically, giving those monoclonal antibodies, the FDA waived any sort of testing for that, so that this could go ahead and save people's life. It's not as dramatic with COVID-19, but we still have significant death rates, in particular in the elderly population. But it's not that only old people are infected and die. In particular in US, we see way more younger people also dying of the disease, healthy young people. So where you cannot say that there is any predisposition there. So this covalescent serum is basically serum from patients that have gone through the disease. So they were tested positive. They may or may not have been on a ventilator, but they managed to cope with the disease and generated antibodies. And so when you do a blood donation from, and the Red Cross, I actually posted that on Facebook, I think yesterday, the Red Cross is seeking particular people who have had COVID-19 and survived to give their blood so that they can do, enrich the serum. And then in the serum, you have antibodies from them. So their antibodies will be used to save other people who have not been able to generate antibodies yet. That will be a short-term quick effect. On the long run, there are several companies working on vaccine development, and there are different approaches to that. One of them is, for example, that so-called DNA vaccines. Technically, it's actually messenger RNA that is injected into your body, and the messenger RNA is directly transcribing viral protein in your body, for example, the spike protein in that case, to which you would generate antibodies by yourself. Now, when you give these DNA vaccines, you still need about 6 weeks to generate good antibodies. That's just because your immune system needs that long to generate that. So in those times, you're not protected really against coronavirus 2. Then there are other approaches that are taken. The classic approaches, you take a non-functional hepatitis virus B, the core particle, and you add, for example, the spike protein of SARS-CoV-2 to it. There is a Swiss, the University of Basel, they have a clinical trial going on with that approach. And then there's a guy who I know, Peter Burkhardt, who has done the self-assembled nanoparticle. He has generated a self-assembled nanoparticle with the receptor binding domain of SARS-CoV-2. He published in 2010 a paper for SARS-CoV-1 with the same approach. And at that time, they saw in a mouse model, I believe, a 70% reduction of infectivity of SARS-CoV-1. So he hopes that the one that he made for SARS-CoV-2 is similar or better. We will hear about that eventually. So those would be the approaches that are undertaken for a vaccine. But all of these approaches will not be available, let's say, in the next 12 months, probably longer, 18 months maybe, because it has to go through FDA approval, and the minimum time frame for even for a fast approval is around a year, and you still have to do the research as well. So that was a long answer to your question.
No, that's okay. I appreciate the detail. I was also thinking, I'm cheating a little bit because I know you work in drug design. If you're presented with an organism such as this, are there really apparent, I don't know, keyholes or something that you would exploit in order to attack it with the drug, or is the methods that you described really the most readily available way of approaching combating this organism?
So
let me just cut in for a second. This will have to be the last question because we're over time. So go ahead and answer, but then
you're running out of space.
No, it's okay.
Okay, I'll make it.
Tell people
I'll make it
5 minutes, and
I'll make it brief. Because that sequence of the first Wuhan strain was released January 8, what happened is that several teams worldwide, I mean, many of the scientists realized that this is potentially a real problem. So a group in London at the Diamond Light Source actually cloned the protease that is needed for the virus to, so I should explain, when the virus genome is translated, it's producing a single protein. And there is a protease inside that cleaves off that protein into the right pieces that the virus can be functional. It's a unique feature. And the idea is basically if you inhibit that protease, you would inhibit making the virus functional. So what they did is they cloned, expressed, crystallized that protease. And on top of that, they solved the crystal structure and then added they did a fragment screening and had 60 hits. All of those structures were available beginning of this month to anybody who wants to work on it and suggest chemical modifications for new drugs, basically. The trouble with this approach is that a small molecule drug will take a lot of effort to get into FDA approval because the bar is much higher compared to the monoclonal antibodies, because you don't know what that small molecule drug might do on the organism. So you need to do 2 small animal studies, 1 large animal studies before you can even start doing safety studies. And so if there is a new drug available, it would minimally take 4 or 5 years is what I would anticipate. Now there's something else to that, and you can do something that's called drug repurposing. So what that is you have known drugs that are used that you can buy over the counter or so, and you just screen them against the SARS-CoV-2. Several of the drugs were identified that have an effect, like remdesivir, which was originally made for Ebola, and it failed there. It didn't have the desired effect, seems to have an effect on SARS-CoV-2. It's also currently undergoing a clinical trial side by side with another drug that's called favipiravir or something like that. It's a Japanese drug that's there on the market, and it was reported to have an effect on SARS-CoV-2. So I would say probably within a month or so latest, we should have results on these 2 drugs that are really reliable. And if they should turn out to be effective and start infecting, we would have something that basically I would expect that the company who currently owns the patent on those drugs, the one is Gilead, the Rivampamir or whatever it was called, remdesivir, sorry. And the other one, the favipiravir, the Japanese company, I would suspect that they would release the patent so that any company can mass produce those drugs to help people who need it. If we don't do that, we will have a huge death toll in India and Africa.
Okay, so I'm getting a lot of pressure to allow a few more questions. There's one on the chat. What are your thoughts of Cepheid testing and also isothermal PCR Abbott's testing? What are the validity of those tests versus traditional TAC COVID tests? How are the tests going to be deployed to third world countries, countries such as Indonesia that has failed to produce rapid testing?
What's the first part of the question?
What are your thoughts of the Cepheid testing?
Cepheid testing, I'm not familiar with that one.
It's a new test.
I know nothing about this, sorry.
Like the satellite.
I'll
Also,
look it up later.
PCR Abbott's testing, yeah. All right.
I mean, the isothermal PCR is basically, it's basically the same as a PCR, but you don't need a PCR machine that cycles between high temperature and low temperature. So it would be something that could be applied in a like rural area, or let's say Africa, India, those places. I don't know how how reliable they are, but I think similar tests have been actually done for Ebola already in Congo. So I would anticipate that potentially it might work well for SARS-CoV-2.
One last question, anybody?
I have a question if nobody else does.
Great.
Yeah, hi, I really appreciate your talk. It's very informative. So our lab is trying to think of protein-based therapeutics, not so much antibody, but protein. And so there's a paper that has been pre-released from the Pentolte group at MIT and Harvard. Mm-hmm. And so they did in silico modeling and found a 23 amino acid peptide from the ACE2, derived from ACE2. And they've been able to show some preliminary data suggesting that it binds with pretty high affinity to the spike protein. Mm-hmm. Do you think that that might be a better base to start some sort of polymeric-based therapeutic approach? Mostly because I don't know what the regulatory structure looks like versus an antibody, peptide, and small molecule.
It's the same, because your peptide is basically in between. I mean, it might be slightly easier because it's more similar to an antibody because it's protein-based. The trouble is the delivery method of such short peptides. I mean, there have been various things for, what are these guys called? Aptamers, which are DNA type things that recognize proteins. And I think peptide approaches would fall into the same area. Now, one problem with peptides is they get digested very easily. So you have to design the peptide in such a way that it's not degraded. There are various people around who know how to do this, like with L-amino acids, for example, if you can introduce them into such a thing. But most likely you will then also change the property of the binding affinity to that spike protein. So it's not an easy answer, or it's also not an easy question, but it's definitely worth thinking about. I think one of the more promising papers that I saw more recently is a group who has been working on SARS-1. I don't recall where they were, but I want to guess Sweden, maybe. One of the or let's say one of the northern European countries. They had 51 monoclonal antibodies against SARS-CoV-1, and they simply screened their library against SARS-CoV-2 to see which of those are neutralizing. And they identified 4 out of the 51, and they are moving forward with that. They already humanized them. So basically, I don't know if you're familiar with the company Regeneron here in New York, actually. They live off humanized antibodies, and that has been already approved by the FDA for certain type of diseases, like certain cancers, for example, are treated with humanized monoclonal antibodies that are attached to certain cytotoxic chemicals. And so probably the FDA approval for a different monoclonal antibody that is just detecting something else is much faster to achieve compared to a novel IND filing, basically. So IND stands for investigational new drug. And even if an antibody is not really a drug, it still falls under IND. Hope that helped.
Absolutely. Thank you.
All right, now I am going to have to close it down. Thank you so much, Jurgen. This was terrific. And I hope to see everybody on Thursday for what's hot now in cosmology.
Okay. Thank you, Jurgen.
See you guys. And here's my beer.
Cheers, Jurgen.
Thank you, Debbie.